Welcome to the October 2017 Nutritionist webinar. I'm Marianne Fessenden from AMTS and your English language host. This monthly webinar series provides technical talks from internationally recognized educators for listeners around the world. Paula Torillo from Cordoba, Argentina translates and hosts a Spanish language webinar. Marcos Neves-Pierre from Lavras University of Lavras in Minas Gerais, and Marcelo Hens Ramos from 3R Lab will join from Brazil. There will be a question and answer period immediately following the presentation. Listeners can submit questions through me, Marcos, or Paula. A complete recording of archived webinars as well as a question and answer session for each will be available on the AMTS website. For those of you who would listen to the presentations while driving, we have converted the videos to MP3 files that can be downloaded to your device for offline listening. You were listening to last month's presentation by Lance Baumgard as a podcast. This month we are pleased to host Dr. Michael Ballou, who is an Associate Dean for Research and an Associate Professor of Nutritional Immunology in the College of Agricultural Sciences and Natural Resources at Texas Tech University. He is also serving as the Interim Chair for the Department of Veterinary Sciences. He completed a bachelor's degree in animal science from the University of California, Davis in 2002. Michael remained at UC Davis and completed a PhD in nutritional biology with an emphasis in immunology in 2007. Michael's research is primarily focused on how nutrition and management influence the health and performance of dairy calves, heifers, and periparturient cows. He has authored or co-authored 55 peer-reviewed articles, 1 book chapter, and 106 scientific meeting abstracts. Michael has received research support from private foundations, industry, the USDA, and the U.S. State Department. His talk today will focus on practices that can be used on farms to improve immunity in dairy cattle. And Michael, I am going to go ahead and unmute your mic. Hi, how are you?
I'm doing very well. How are you doing?
I'm very well. I'm going to turn the presentation over to you, if I can get it down that far.
Let's see if I can do it like we practice.
Yeah, that's really nice.
It's amazing how much you can forget in 2 weeks.
I know, and I really appreciate that you did everything so early for me. So you should be in control now.
Okay. All right. I guess we're good. I was just trying to quick find the laser pointer again.
Okay, let me make sure that's working for you.
There we go. The arrow, I guess.
And right now, I'm still on my introductory slide, as long as you know that. I think you didn't have control when you forwarded.
Yep. Okay.
All right. Good. Now I'm seeing what everyone should be seeing.
Great.
All right.
Well,
I'm going to get off the line, and you can take it away.
Sounds good. I looked at my slides, and I got a lot of slides, so I got to get going, I guess. So thank you again, Marion, for the introduction, and looking forward to the opportunity to share some data and some thoughts I have on, how to improve the health of, transition dairy cows. So some expected outcomes. I want to talk a little bit about immunological phenotypes of transition cows. Part of my lab, we want to know what a quote-unquote good immune system looks like. That's not an easy question to answer, and that'll probably be apparent as I walk through some of this, but I want to talk about, kind of what we know about the immune system of transition cows and what kind of increases their risk for disease. I want to talk a little bit about stress, because we know that stress plays some role in the etiology or the risk for developing infectious disease during the transition period. And I want to talk about the actual potential impacts of stress on immunity. And go through, what are the different types of stressors an animal can experience, what are some of those potential impacts of individual stresses or combinations of stressors on the immunity of transition cows. And then lastly, I also want to consider some alternative pathways explaining increased risk for transition disease. So trying to differentiate between stress versus maybe indirect pathways of increasing the risk for disease. So, I mean, the reason why we're having this talk is that we know that there's a high incidence of both infectious and metabolic diseases during this transition period. We also know that this isn't mutually exclusive. So an animal that, for example, that gets mastitis is more likely to get hypocalcemia or develop a ketosis or a DA, and vice versa. A cow that gets hypocalcemia, we know that an increased risk for like metritis and potentially mastitis. So again, it is complex and it's not mutually exclusive. So what I want to get into is talk a little bit about the immune system of a cow. If you broadly look at the immune system and what the immune system in any animal is there for, its primary role is to recognize self from non-self. The self we don't talk a lot about, but it is really important that the cow's immune system actually recognizes self and doesn't respond to it. But it also recognizes non-self things when an animal becomes infected with a non-self microorganism. So therefore, the immune system's really role is to protect the host from non-self microorganisms, whether it's a virus, a bacteria, a fungi, or a protozoa. The immune system is if an animal's been exposed and it's colonized and it's entered into a tissue, that the immune system recognizes it and says, hey, this shouldn't be here. Let's try to eliminate this. So why is it called an immune system? Because there's lots of different components to it. I once had a grad student that wanted to measure every aspect of the immune system. I told her, I said, I don't have the time and money to do that. I said, we need to pick a few things to measure that kind of encompass the immune system, the different layers of the immune system that we'll walk through in a little bit. But on the other side of things is you can't just measure one thing and get an idea of what's going on, because there's lots of different redundancies and synergies and different things going on with the immune system. So there's lots of different layers to it. We'll kind of walk through some of the layers, and hopefully you'll get a better idea of how the immune system is kind of generally just this very rudimentary kind of. So again, there's lots of different layers, various redundancies, and many different interactions, which makes it difficult to understand and study it sometimes. And I like to talk about this, and I'll show a little bit of data from mice that a breakdown in any component of the immune system may increase the risk for disease or actually have no effect at all. So I thought a nice study that was done in mice, and I'd love to do this in cows, was looking at risk for mastitis. So they induced E. coli mastitis in a mouse model, and they gave an intramammary E. coli challenge. And what they did was they took a group of mice and pharmacologically depleted a certain type of immune cell. And we'll talk about this immune cell a little bit in a few slides, but it's called a macrophage. It's an important immune cell leukocyte that primarily is in tissues and is looking for non-self things that shouldn't be there. So when you infuse this E. coli into the mammary gland of this mouse, these macrophages should recognize that this shouldn't be here, and they should really set off a cascade of signals that's going to recruit immune cells to the mammary gland that should, and other things, but it should eventually remove the E. coli from the mammary gland. And in this study, when they pharmacologically depleted those macrophages, they saw no effect on neutrophil recruitment. So the neutrophils were still recruited. And they saw no difference in bacterial growth than animals that didn't have those macrophages depleted. So again, the study suggested that the macrophage in this case wasn't really depleting the macrophage, didn't really have any effect on the pathogenesis or the response of these animals to that E. coli challenge. What they also did is they depleted another type of immune cell called a neutrophil. And neutrophils, again, we'll talk about them in a little bit, but they circulate around blood, and then once they get a signal, primarily from macrophages and other cells, those neutrophils will get recruited to the site of infection. And their job is basically to go in and efficiently clean up and eliminate various pathogens, primarily bacteria or bacteria, in this case, this E. coli. So when they depleted the neutrophils from these mice and then infused them with E. coli into the mammary gland, there was uncontrolled bacterial growth. So bacteria just rapidly grew in the mammary gland or in the milk. there was tissue damage, animals became septic, and there was a high risk for mortality. So this is just kind of showing that, a breakdown in any component, in this case, or in this study, removal of macrophages really had no effect on the pathogenesis of the disease. In contrast, neutrophils are really important in the response and protection of the mammary gland to this E. coli mastitis. So let's get into the kind of the basic framework of the immune system. We'll start and we'll walk through this, but the initial part that we'll talk about are the physical barriers of the immune system, and then we'll get into more specialized leukocyte functions that are very specific and do something very specific to either limit the growth or to kill a invading pathogen. So the first layer of the immune system is the physical barriers, and this is really the primary protection, and we often forget about these things and how important these things are. I like to use analogies to describe the immune system, and typically what I tell students is, if you're trying to design an immune system, it's like designing a state-of-the-art home security system. And so that's kind of, what we're going to talk about. And protecting your internal organs is like protecting your house. So all your organs are extremely important, like your wife and your kids and everybody that lives in your house is really important. You want to protect them. And so the primary protection of these physical barriers is just limiting access to those internal organs. So the skin, a closed teat end, doesn't allow anything to get in. You can't get mastitis unless something gets inside the mammary gland, and that closed teat end, the teat end or teat canal keratin, and all these mucosal barriers. We're constantly breathing in bacteria and viruses, and I'm fighting a little bit of a cold right now, and both my kids have been sick this last week, so my house has got a lot of stuff in the air right now, but I'm constantly breathing in stuff, and there's these mucosal barriers in my respiratory tract, and I'm constantly consuming food and eating things and consuming bacteria in food, and there's these mucosal barriers that are really forming a protective barrier and a physical barrier from preventing these organisms that I'm being exposed to, constantly being exposed to, from gaining access into my body. So the analogy I use back to the home security system is it would be like a large fence protecting, like a perimeter fence protecting your property, so they can't even get close to getting to your organs. If a relatively small number of microorganisms infect a competent immune system, so an infection means that there's actually, you've been exposed and there's some colonization of these microorganisms, but if a relatively small number of microorganisms infect a competent immune system can actually eliminate that threat without causing any signs of clinical disease. So a good example of this is looking at coliform mastitis in cows. So if I take some cows and 150 days in milk and I grow up some E. coli in my lab, a certain E. coli, and I infuse 100 colony forming units of this E. coli into these cows at 150 days in milk, the majority of those cows, almost 100% of the time, those cows will clear that infection without any signs of disease. You would never know that those animals were exposed to that E. coli. They wouldn't develop mastitis. You wouldn't see any clinical changes in really the behavior of these animals, and you may or may not see a fever in those animals. However, if I give that same dose of E. coli to cows at, let's say, 10 days of milk, we'll see a lot of those cows will actually develop mastitis, and we'll see some of those cows will develop really severe mastitis. So there's more going on in these transition cows that the physical barriers have actually been compromised. There's some other things going on here, so that's where I want to kind of go and look at what are the leukocyte phenotypes and all these other aspects of the immune system look like in early lactation that increase the risk for that these animals are going to develop infectious diseases like metritis or mastitis. So a secondary protection are antimicrobial secretions. So on those mucosal surfaces I talked about, I study a lot in the gastrointestinal tract, but the gastrointestinal tract has a lot of antimicrobial secretions there. There's a lot of small peptides that you can almost think of as like natural antibiotics being produced by certain cells in the gastrointestinal tract that kind of coat those mucosal surfaces. So it's really not a great environment that a lot of potential pathogens want to colonize. So the analogy I use would be something like a Rottweiler or a pit bull inside your front fence. I've included Rottweiler there because I used to just have pit bull and I offended somebody, so I included a Rottweiler. It just looks scary. Okay, and there's enough data to actually show if you have a dog in your background, you're less likely to get robbed. So I'm comfortable with this analogy, and I'm not trying to offend anybody. But Rottweilers are scary looking, and pit bulls are, I think, scary looking too. But this would be essentially, if something got through that perimeter fence, again, it's not going to be an environment that is really conducive to growth of that pathogen. The third layer is again, we're going to revisit that macrophage cell. So again, I had talked about a little bit ago when we were talking about the mouse mastitis model. And these macrophages, again, live in tissues, and they're they're phagocytes, and they're constantly looking for something that shouldn't be there. So they're looking for non-self antigens. And when they get exposed to these things, they signal an alarm. So the analogy I would say is if somebody opened your front door, they got through the fence, they got past the dogs, and they've actually now opened up the front door or broken a window and are getting close to getting into your house or actually in your house. So again, these macrophages are living in tissues, which again, your immune system is trying to protect these tissues. So you're getting really close. So these macrophages recognize, okay, we've got a big problem here. This shouldn't be here. So part of what a macrophage does is they signal, and I like to think of them as an alarm. So this is just a little picture of a macrophage. This would be a bacteria. Let's just say this is an E. coli that we infuse into the mammary gland. This macrophage that's living in the mammary tissue recognizes it, it phagocytoses it does some things with it. But one of the things that I want to point out is it just produces these pro-inflammatory mediators. So one of the things that macrophages do is they're really good at stimulating inflammation and basically saying, we have a problem, let's get some help over to this site of infection. So when you look at transition cows, early lactating cows have an increased inflammatory responsiveness to lipopolysaccharides. So there's been a few studies. I've done some of this, and Lorraine Sordillo showed this back in 1995, that there was an increased secretion of pro-inflammatory cytokines from macrophages cultured in vitro when they were stimulated with LPS. So they just had the propensity to produce more. Another nice study that was done in the early 2000s was they did a crossover design. So cows, when they were both in early and in late lactation, half the cows got it when they were in early, and half the cows got it first when they were in late lactation. But every cow got LPS challenged both in early and in late lactation. But what they saw was when they challenged those cows in early lactation, LPS intramammary, they saw that the local and systemic inflammation were more pronounced when they were in early lactation. Again, given a fixed concentration of a purified pathogen-associated molecular pattern, or just seeing how much, they're basically looking at how much inflammation can these animals produce. And when those cows were in early lactation, they had a higher capacity to produce more inflammation. And so I often get asked, and this is a valid question, and I don't have a good answer for it. So is how much inflammation is enough inflammation? I mean, that's just a good biological principle. We know that biology responds quadratically, and usually some is good and more often at some point will become detrimental. And inflammation is exactly the same way. Is you need some inflammation. You want to have some inflammation when you're infected and upregulating this immune response, but you also can swing the pendulum way too far and actually have way too much inflammation. And so what I typically say is I want the amount of inflammation to be proportional to the threat. And so if I see somebody breaking into my house or into my backyard and trying to steal my barbecue, yes, I like my barbecue. Can I live without my barbecue? Yes. So if I see a guy and he's not threatening the rest of my house, I'm probably going to call the Lubbock Police Department, but I'm not going to call the Air Force Base out of Abilene, Texas, to come and send fighter jets to my house to drop bombs on my house. That would be an analogy that's showing a way over aggressive or in proportional response to the threat. And again, dropping bombs on my house solved the problem of stopping the guy from killing or taking my barbecue. But what it also did is it caused a lot of collateral damage, killed me and my family and probably some neighbors and everything. So I hope you understand inflammation can be in your immune system, if directed against the wrong thing or too aggressive, can cause a lot of damage. So like most biological processes, the counterbalance of the anti-inflammatory response, especially after an inflammatory response, is extremely important in kind of counterbalancing and keeping things kind of in homeostasis. The last layer of the immune system I want to talk about is the neutrophil, because this is a cell type that's been studied a lot, especially in cattle. And I call it the fourth layer of protection, because it really is that cell that it circulates around blood, and it's getting recruited to a site of infection. It doesn't, unless you have a bloodborne disease and we're infected directly in the blood, it's most likely going to be another immune cell that sees the invading pathogen first, and it's going to tell neutrophils that they need to come there and take care of it. So the analogy I use for neutrophils is like law enforcement. So again, they're driving around, and when they get a signal or a call, they come to the site of infection. Along with neutrophils getting recruited to the site of infection, there's lots of other humoral things in blood and in extracellular space that are also getting recruited and upregulated there, that are going to help in kind of controlling and keeping that infection local and eventually, hopefully, clearing that infection. So the neutrophil, they're again a phagocyte. So this would be like this little yellow thing over here being like an E. coli. When a neutrophil sees that E. coli, it phagocytoses it, and one of the things it's going to do is either digest this thing in a phagot lysosome, or it's going to produce an oxidative burst. Essentially what it's going to do is it's going to kill that bacteria, hopefully, is the main job of what a neutrophil is. A neutrophil really is more than being like law enforcement. It's more like a kamikaze mission. They go into a tissue, they try to kill the invading pathogen there, and then they ultimately kill themselves. So, like I said, neutrophils have been studied quite a bit, and especially during the transition period, and by lots of different groups. And it's well documented that many neutrophil responses are suppressed. And even though we're in 2017, these two reviews in 2003 are great reviews on neutrophil responses during the transition period. Basically, there's suppressed number of mature neutrophils. There's suppressed chemotaxis. Chemotaxis is the ability to get to the site of infection. So again, they're coming from blood to tissue. There's suppressed phagocytic capacity. So this would be like a cop, the chemotaxis would be like a cop driving on flat tires. Suppressed phagocytic capacity, the analogy I would use is they drop the handcuffs or have a hard time getting somebody put in handcuffs when they come in contact with them. And they actually have suppressed oxidative burst and total killing capacity. So almost every aspect of the neutrophil is suppressed, can be suppressed in early lactation. So this is the kind of the analogy. When I think of a neutrophil in early lactation, I think of Barney Fife. So if you know who Barney Fife is, that makes sense. Again, it's just they're really a dysfunctional, they can be a dysfunctional neutrophil in early lactation. So why would suppressed neutrophil responses increase risk for disease? Because if we go back to that mouse model where they did a mouse mastitis model, it showed when they knocked out the neutrophil, there was uncontrolled growth of that E. coli in the mammary gland, and it ultimately resulted in high tissue damage, sepsis, and a high risk for mortality. And so again, thinking of neutrophils like law enforcement, they keep the peace, limit any uprising before they can reach a critical threshold that you really start getting some damage and some traction by that microorganism. So I have a working hypothesis is that there's really so what does the immune system of an early lactating cow look like? My working hypothesis is that there's a potentially an uncoupling of the inflammatory response. So I talked about that data showing that cows in early lactation, they're really on edge and can produce a lot of inflammation. But when you have a suppressed other innate immune defenses, that really is a dysfunctional immunological phenotype. Because again, that inflammation can cause a lot of damage. And when you couple that with suppressed innate immune defenses, it may cause this to just go unchecked, and that's why you see such severe disease, or can see such severe disease in early lactation. So here's a study that we just wrapped up. It's an unpublished paper, and we actually haven't even submitted this into an abstract form yet. But we enrolled 75 Jersey heifers and cows, so there were 36 heifers in the study and 39 multi-pairs cows in a prospective longitudinal study. And we enrolled those cows at -7 days in milk relative to expected day of calving, -7 days relative to expected day of calving. We took a blood sample from those animals at -7, -3 relative to expected day of calving, and then at 7 days of milk and 28 days of milk. And primarily, the student was focused mostly on just looking at neutrophil functionality, a few measures of neutrophil function. And a couple things they looked at is they looked at some adhesion molecules. So remember, neutrophils are trying to get from blood to a site of infection. And somehow, one of the ways that they're doing this is they have these adhesion molecules on their surface that allow them to kind of attach loosely to the vascular endothelium that allow them to eventually get into a tissue. And then once they're into a tissue, again, one of the ways that primary ways that they can kill a pathogen is by producing this oxidative burst. And so we looked at, and I'll just present that data, the surface adhesion and oxidative burst to an environmental E. coli. We recorded a lot of health outcomes, and the ones I'll report here are we looked at clinical mastitis, vaginal discharge at 28 days of milk to look at endometritis, pregnancy at 150 days of milk, and then I broke up choline as a class variable saying less than 28 days of milk or cold greater than 28 days of milk. And basically we wanted to look at were the neutrophil responses around calving different dependent on whether or not they developed mastitis or these other health outcomes. So just looking at these figures, and I'll kind of orientate you to these because the rest of the next 4 or 5 slides will be like this. So I'll have the leukocyte response or the neutrophil response measurement on the y-axis, and then time relative to calving on the x-axis. And then I will be presenting the data broken up, whether or not they had the different no health outcome or the health outcome that we were specifically looking at. So for example, neutrophil L-selectin, so an adhesion molecule. And so time relative to calving. So cows that got mastitis versus no mastitis, you see really no difference in their neutrophil L-selectin response during this period. However, when you start looking at neutrophil oxidative burst response on the y-axis, so this is that neutrophil oxidative burst, and again, mastitis versus no mastitis. So the mastitis group is in the checked line, and in the solid line, excuse me, the solid line is no mastitis. You start seeing some differences here. So even at our baseline sample, cows that developed mastitis, and again, cows were developing mastitis, and predominantly if you go back and look at the health records, most of the mastitis was occurring in the first 4 weeks of the lactation. But you can see even at our first blood sample at enrollment there, cows that eventually developed mastitis already had elevated neutrophil oxidative burst. We're almost hypothesizing maybe that these animals were, there was already some exposure, and that way this response is already being stimulated a little bit. And so some of it could just be that these animals that develop mastitis, obviously, they may have already been exposed out here. But what's also interesting is you see at 7 days of milk, these animals that develop, these cows that develop mastitis had a suppressed neutrophil response at 7 days of milk. Again, kind of corresponding with the high incidence of, or the timing of the high incidence of mastitis at this period. Again, I'm not going to get into the cause and effect here, but I'm just showing you some associations that we saw. So this is looking at endometritic animals diagnosed at 28 days of milk based off of vaginal discharge. So we classified the animals as non-endometritic, so either no vaginal discharge or clear discharge. Mildly endometritic there was some discharge, and I'm drawing a blank exactly what percentage of it was cloudy. And then we had severely endometritic cows, which was purulent, so it had a foul odor to it and blood in it, and had more than 50%, I think, mucus in it. So again, we diagnosed those animals and then went back and looked at this. And so again, prior to calving, we saw really no differences in this neutrophil L-selectin, so the adhesion molecule. But out here at 7 days of milk, we saw that animals that were not endometritic, had the lowest L-selectin. Intermediately was the mildly endometritic, and then the severely endometritic animals had the highest peak at 7 days of milk. This may just be a reflecting the exposure of these animals at this time. And so again, we don't know exactly, but again, showing that there is some differences in neutrophil response of these animals that are, and again, these are being classified out here, and we're really seeing no difference out here at 28 days of milk. We saw the differences back here at 7 days of milk. Looking at neutrophil oxidative burst, no statistical difference. There is, we had quite a bit of variation here. This is interesting to me, and we're following up on this and actually in the process of designing a more comprehensive assessment in a larger data set. But what's interesting to me is that these severely endometritic cows, the non and mildly endometritic cows had an elevated neutrophil oxidative burst prior to calving, and then kind of had come back down and flattened off. Whereas those severely endometritic cows, although not significant at -3 days, just remain flat throughout.
Dr.
So this is, yeah.
Sorry to interrupt. I've had a little bit of feedback that perhaps you're speaking a little too softly, especially towards the end of your sentences. So apparently it's hard to hear.
Okay. I'll try to speak up. Let me check my microphone here. I'll turn it up a little bit. All right. So moving on to the next, and this I want to kind of get through this a little bit because I got some other stuff I want to talk to. This is looking at culling. So what I did is I took animals that were not culled. So this is these triangle bars here. And then I took animals and I said they were culled less than 28 days in milk. So in the solid black line. And then I have animals that were culled greater than 28 days in milk. And when we look at neutrophil L-selectin, we see no differences in these groupings prior to calving. But what's interesting is when you look at 7 days of milk, these animals that were culled less than 28 days of milk had reduced neutrophil L-selectin expression on the surface at 7 days of milk. And then what's interesting is these cows that were culled, or these animals that were culled greater than 28 days of milk, look at their neutrophil L-selectin out here at 28 days of milk. So it's come up and it's stayed up out here at 28 days of milk. So again, these animals are more likely to be culled at 28 days of milk. And when you look at neutrophil oxidative burst, we don't see as significant, but we see some similar trends here. So these animals that were culled less than 28 days of milk, again, numerically, these animals have suppressed or lower neutrophil responses at 7 days of milk. And so if I just go back to the previous slide, you'll see, again, it's consistent with this other neutrophil measurement that we were looking at. And then what's also interesting is that these animals that were culled greater than 28 days of milk, again, we're seeing that they're up and they continue to kind of persist up. So again, if we go back to the previous slide, looking at neutrophil L-selectin, these animals that are getting culled greater than 28 days in milk, they go up and then they stay up. Those responses stay up. So these leukocytes are active, which may not be a good thing in this. So what does an ideal immune system of a transition cow look like? And if I had to simplify it, I would say rapid upregulation when it's needed. So if a tissue is infected, you want to see the immune system rapidly upregulate. But what you also want to see is that response is proportional to the threat. You don't want to see an overreaction of any type of response. Neutrophils you get too many neutrophils into a tissue, you're going to cause a lot of damage. We've been studying neutrophil recruitment into the gastrointestinal tract, and it's seen that when you get a lot of neutrophils recruited to the intestinal tract, you see a lot of histological, morphological changes that are consistent with damage. So you want that response to be proportional to the threat. And then what you want to see rapid downregulation once that threat is removed. So again, rapid upregulation, rapid downregulation. You want that, the analogy I tell people is if somebody breaks into your house in the middle of the night, you call the law enforcement, you want them to get to the house as fast as you can, okay? And you want it to be proportional to the threat. We've already talked a little bit about that. And then you want them to leave, okay? They take care of the problem. You don't say, hey, why don't you guys spend the night? I'll cook breakfast for us in the morning. You want them to get out of there because these immune cells, again, can cause a lot of damage. So you want to downregulate them kind of clean that tissue up. So that's what an ideal immune system looks like in these cows. And we know that there's a lot of holes in breakdown in a lot of different layers in the immune system. There's holes in the physical barriers of the immune system that increase the risk for disease. There's changes in these different leukocyte population that's increasing the risk for disease. And again, my working hypothesis is that we have, a propensity to produce a lot of inflammation. And when you couple that with a suppressed other aspects of the innate immune system, that really increases the risk for disease, and it really increases the risk for severely clinical disease with a lot of tissue damage that may end up resulting in animals getting cold. We've shown some data, and I didn't put in this presentation, but animals that have inflammation and early lactation are more likely to get cold and have decreased reproductive performance in subsequent lactation, and that's been also shown by other groups as well. So now what I want to do is spend the next 25 minutes, or the second half of the talk, talking a little bit more about what are the mechanisms resulting in this dysfunctional immune system. And I really want to kind of talk about stress versus alternative pathways. So the term stress, we use it a lot, and we use it in a lot of different contexts. And unfortunately, it becomes very vague, and sometimes we overgeneralize what stress is. And when I usually ask people, I said, and when I talk to students, I say, what is the first thing that you think of when you hear stress? Most people think negative, okay? Most people think negative, especially when you think of disease. I've been studying for exams, and that's why I'm got this nasal discharge now. Okay, I've been stressed, and I'm higher risk for disease. But the thing that people actually forget about is stress is natural, and it's a physiological response that promotes a response to threat or change. I will tell you, we have knocked a stress response out of some animals and seen some very negative things relative to that. And I'll also tell you, if we were all sitting in a room and a bear or a lion walked in the room, I hope my stress response kicks in because I want to get out of there, okay? And so again, stress can be good and it can be bad. And so let's kind of try to delineate a little bit, maybe what makes a stressor bad in situations where stress is good. I often joke that I want my grad students to have some, stress, because they're usually active in doing things if they're in a stressed situation. They always say forced and focused work are where you're the most productive. The problem is most of us have to work with forced work now, and so you're usually stressed and you're at a height. And so again, I'm just trying to illustrate that stress can be good, and it is a natural physiological response. And I sometimes think we exaggerate the negative effects of stress, especially with respect to transition cow disease. But I also will show you and admit that stress plays probably both an important role in the adaptation to lactation, that we want that stress response because there are changes going on. That's why we call it the transition period, because there are changes going on. So we do have adaptation going on. So we do need some type of stress here. But under certain instances, it does increase the risk for disease. Okay, so we'll go through some of that. We've known about the effects of stress on the immune system since 1936. It was done in mice where they did psychological and physiological challenges to mice, and they observed that their lymphoid tissues got smaller. They had stomach ulcers and then adrenal gland enlargement in the animals exposed to chronic physiological and psychological stressors. So what's the difference between chronic versus acute? And I even can classify as repeat acute stress. But to me, the difference between chronic and acute is really deals with the ability to adapt. If an animal can adapt to a response, I would consider a stressor. If it's a change and it is a stressor, I would consider that more acute. Where if it becomes they can't adapt to that, I would consider classifying it as a chronic stress. So what stresses a cow? Basically the same thing that stresses us. So why are cows creatures of habit? Well, cows are creatures of habit because they have control. Okay, and the less control an animal has, whether or not it's you, whether or not it's a cow, the more stressful. I am a creature of habit myself, and when I'm out of my habits, I increase my stress levels. So the change and switch from non-lactating to lactating, like I said, is the transition. There's change. It's abrupt and it's dramatic. And we typically know if you want to make changes, you typically want to make them slow. But unfortunately, the switch from non-lactating to lactating is abrupt and it's dramatic. It's a big change. So typically, bigger changes have the ability to be more stressful than smaller changes. So we already see that the cow, possibly a lot of cows may have some negative impacts of stress during this transition period. A good example of there's lots of different laboratory stress models, but the one I always, illustrate, and I think it paints a clear picture, is, again, if we were all sitting in the same room and I had a loud buzzer go off, and it just kind of kept going off, then it turned off, so it went off for 10 or 15 seconds, and then it turned off for 5 minutes, and then in 5 minutes it went off again for 15 seconds, and it turned off for 5 minutes. If it was very repeated, it would initially stress us out, but eventually, once we figured out that it was going to go off for 15 seconds, and then it would turn off for 5 minutes, we again had the control, so it's more of an acute stressor. However, if you have that buzzer just go off randomly, and the animals can never adapt to it, that's a great model of chronic stress. My brother's a dairyman, and we were talking once, and again, this actually just came up again recently when I was talking with another dairyman, and they were talking about the way they fed their calves and the way they weaned their calves. And basically they said whenever we run out of milk, that's when the calves get weaned. So when they start feeding milk, they feed it to the youngest animals, and then when there's no more bottles left, those animals don't get milk, they get weaned. But what happens is some days those animals are getting milk, some days they're not. They never know. I just said it's better that they know that they're not going to get it, and that weaning can be stressful, but you pull them off of it, and then it's like pulling a Band-Aid off, and then it's best to be consistent with your responses. That's why cows and animals crave consistency, because it is all about control and not changing. So generally, we can classify stress as either psychological or physiological. And there's some overlap in some of these, but I'll just kind of broadly classify some of these, like psychological stresses that a cow may be exposed to, include interactions with both cows and humans, pen moves, stocking density, handling, novel environments. So if they've never been exposed, first time heifers are going through a parlor, that's going to be stressful. Changes in weather, okay, that can be both physiological and psychological. And then macro and micro environments. So macro being, more changes in the weather, but micro environments being, especially like, for example, in a freestall barn, you may have micro environments in a barn that may create some competition over some space there. Any variation in daily routine. I always tell my grad students, for example, when we do a calf study, I said, I'll give you some parameters I want you to follow. If you want to feed at 6 in the morning, that's okay with me. If you want to feed at 7:30 in the morning, that's okay. I'm going to have some interval between the feedings that you're going to have to wait, but whatever you decide, I want you to feed at that time every single day. So again, I want things to be very consistent. So just looking at some of the effects of psychological stress, looking at some behavior and immunological endpoints, pen moves can alter an animal's behavior, and it can persist up to 3 to 7 days. Dry matter intake also decreased when a single cow was introduced into a stable group of 11 cows. Dominant lactating cows did not have changed behavior, but the intermediate and subordinate cows produced 3.8 and 5.5% less after a pen move. Overstocking, so whether or not you classify that as access to feed and or a comfortable place to rest and or space, creates competition for available resources, whether or not it's feed, just space, a place to lay. Reducing feed bump space from 1 to a half a meter per cow increased aggressive behaviors, and subordinate cows reduced time eating after feed delivery. Increasing stocking density also decreased lying time among subordinate cows. So again, you're seeing kind of a thing where there's a lot of negative impact on subordinate cows. And really, anytime you increase competition in animals, you can create an opportunity for winners and losers. And so again, some of these losers are going to have chronic stress and potentially could have an increased risk for infectious disease. So here's a study that, and it's really a review, but with some new data reported into it. So looking at social stressors and the effects of immunity and health on periparturient dairy cows. So this study was done at the University of Minnesota when Ricardo Chabel was still there. And what we actually found was dominant cows were actually more likely to have uterine disease and be culled from the herd. So I'll just kind of go through this real quick. This is the probability of uterine disease. This is the number of interaction that a cow had in a day, and then this is the displacement index. So it ranges from 0 to 1. So displacement index is looking at the number of aggressor reactions. The displacement index is the ratio of aggressor being the aggressor versus being the one that was displaced. And so if you have a ratio higher to 1, or the denominator is the total number of interactions. So being closer to 1 means you're having a lot of interactions that are, or the majority of your interactions are aggressive interactions. So what's interesting is you see there's a probability, there's an increased probability of getting uterine disease when you have a large number of total interactions, and most of those interactions are being dominant. And this is looking at, that's probability of uterine disease. So this is probability of being removed from the herd. Again, you're seeing that same trend. A lot of interactions, and a lot of those interactions are aggressive interactions. And so, yes, I think being on either side of the spectrum, being subordinate and or overly dominant, can be negative. So here's just a diagram of a couple different scenarios. So here's a subordinate cow right here, and she's being displaced by this cow in the checkerboard. Or sorry, this checkerboard has just been displaced. So when you have stocking density less than 100%, this cow, if she's submissive, she left but is able to find another bunk space, she's probably going to be okay. If you have the same submissive cow, but you're overstocked, she's going to spend a lot of time trying to get in, but is not overly aggressive. And she may still eventually find some bunk space, but she's going to have a lot harder time and maybe spend less time at the feed bunk than if you weren't overly stocked. And then you have the submissive cows. Even if you have a low stocking density, these are really submissive cows. They get displaced, and then they don't even try to go back to the feed bunk. And then you have these overly aggressive and dominant animals like this checkerboard down here that displaces this cow, and then she moves over here, and then she displaces another cow, and then she moves over here and displaces another cow, and eventually she finally eats. But she's basically spent most of her day or time establishing her dominance over the animals and hasn't spent time eating, which will eventually translate into milk production. So again, like I said, either side of the spectrum, being overly submissive or overly dominant, is probably a negative phenotype in terms of temperament of these animals. So for some physiological changes, parturition, there's a lot of change going on. Demands for nutrients and energy increase. And parturition itself is stressful. There's a lot of things going on. We know the metabolic demands of lactation increase. A great study that was done up in Iowa, by the USDA group in Iowa State up there, where they mastectomized some animals and left some animals intact. And they both went through parturition, and they quantified a lot of different leukocyte and immune responses on these animals. And if I just generalize here, about 2/3 of the leukocyte dysfunction was attributed to actually having the mammary gland. So it more is the metabolic demands of lactation than going through all these pin moves and the actual act of parturition itself. So again, 2/3 of the leukocyte dysfunction is actually attributed to that this cow produces milk and all these physiological changes going on associated with that. So we know energy demands increase. Reported that portal drain viscera and blood flow and oxygen consumption by the liver doubled during the transition period. These animals are consuming a lot more nutrients, and they're producing a lot more energy, and they're having to transport nutrients around and generate a lot more ATP and synthesize a lot more things. So there's just a lot more demand on these tissues. And just imagine, that this is a big physiological change taking place in these animals. And that stress response associated with this is important to allowing those physiological changes to take place, but they can also increase the risk that these animals will get infectious disease. Oxidants. Okay, so these oxidants can either come directly from metabolism, or they can be coming from leukocytes associated with that oxidative burst that those neutrophils are producing. But the point is we have an increase in the production of these free radicals, or these oxidants, and we can get depletion of some of our antioxidant systems and actually can increase oxidative stress in these animals. Metabolizable protein or specific amino acid requirements may increase during early lactation, and there's some data to suggest that the immune system likes to have certain amino acids for certain leukocyte responses. Calcium demands increase at calving, and many cows are subclinically hypocalcemic until bone resorption fully initiates. And so, blood calcium levels drop, and this is beyond just becoming clinically hypocalcemic, but now we have a better understanding of these subclinically hypocalcemic animals and potentially what this is doing with the immune system or certain things with the immune system. So looking at that, so this is data from Jose Santos's lab down in Florida, and cows with subclinical hypocalcemia, so had reduced neutrophil oxidative burst. So this is looking at neutrophil oxidative burst, again, day relative to calving. And again, you can see cows that were subclinically hypocalcemic had reduced neutrophil oxidative burst at both 1 and 3 days post-calving. So most people don't care that much about neutrophils, but if you look at incidence of metritis, we see that those subclinically hypocalcemic animals, about 64% of those animals were subclinically hypocalcemic, where the normal calcemic animals, only about 16% of those animals had metritis. So when you do the odds ratio, animals were 3.2 times more likely to get metritis if they were subclinically hypocalcemic. And again, that ultimately resulted in increased days to confirm pregnancy. It's estimated that up to 50% of cows are subclinically hypocalcemic when fed a non-acetogenic diet, and I've even seen higher than 50% when they're fed some acetogenic diets. And data tend to suggest that the distribution shifts to the left, so you should have less subclinically hypocalcemic animals when fed an acetogenic diet. So going back to the oxidative stress, so again, we talked about increase in the oxidants in early lactation, both metabolic and leukocyte derived, but you can have that increase, and what that can ultimately do is can deplete the antioxidant systems. And Weiss has done a lot of research looking at different antioxidants and shown that a lot of these antioxidant systems can become depleted in early lactation in these animals, and it probably has to do with the accelerated production of these reactive oxygen species. And when you look at these animals when they get in oxidative stress, and you look at the leukocytes and the immunological phenotypes of cows under oxidative stress, you see it looks a lot like an early lactation cow. They have an ability to produce higher levels of pro-inflammatory cytokines. You see suppression in other immunological defenses, including neutrophil function. So oxidative stress looks a lot like the immunological phenotype of cows in early lactation. And many vitamins and minerals are either directly antioxidants, like vitamin A, vitamin E, vitamin C, or they participate in antioxidant systems. So a lot of the minerals, so for example, zinc, manganese, copper, are playing important roles in these superoxide dismutase or these antioxidant systems. So, when we look at some of these micronutrients and specifically kind of focus more on the antioxidant systems, are those 2001 levels sufficient? And I guess we'll find out. And so vitamin E, was increased in 2001 because of that data showing that it was improving neutrophil function and decreasing incidence of mastitis. So this was the first nutrient that the recommendation was based upon a health outcome, which is kind of the philosophy that human nutrition has moved to, and I suspect that animal science will subsequently kind of move a little bit towards that direction with at least with respect to some nutrients. 1,000 IU during the dry period, 500 during early lactation. Some research shown improvements when you supplement higher levels, 2,000 to 4,000 IUs during the transition period. Selenium, FDA regulated 0.3 parts per million. It is pretty well known that organic sources. I get asked a lot, should I do all organic? Should I do half organic? Should I do a third? And I see it under the sun. Everybody, depending on the nutritionist I talk to, they have their special formula or ratio of organic to selenite sources that they deal with. I know, and I don't have time, but I can tell a story about organic sources increasing blood selenium levels and glutathione peroxidase activity when fed organic sources. Vitamin A, most of the data don't really support feeding higher levels than what was recommended with respect to leukocyte function or mini health. Zinc, again, most of these minerals, severe deficiency, you'll see reduced disease incidence, and increasing sometimes you'll see improvements in health. So here was a study done, increasing dietary zinc from 40 to 65 parts per million, reduced somatic cell counts in an inflammatory marker in that study. When we look at copper, again, marginal copper deficiency can reduce disease incidence just like zinc did. You may consider feeding higher levels of copper, especially if you're having some antagonistic, high levels of molybdenum, or have high levels of iron in your water, for example, because again, a lot of these minerals are competing with each other for absorption. Most diets contain around 10 parts per million. So again, without any antagonists, you may supplement, 5 to 10 parts per million to those animals. Heifers fed a low copper diet, so again, below that, recommended 10 parts per million of 6.5 versus 20 parts per million from dry to 42 days of milk had reduced response to intramammary E. coli. So again, that marginal copper deficiency reduced the health of those animals when they were exposed to a E. coli challenge. Manganese is something we may see increase. More recent data suggesting that this might be too low, the requirement may be closer to the 30 to 50 parts per million, and magnesium toxicity isn't a major issue. So again, just kind of in conclusion, I got 3 minutes to kind of go through some conclusions and wrap some of this up. The etiology of the immune dysfunction, or why these transition cows have an increased risk for disease, is complicated and multifactorial. And that's why I titled this talk a systematic approach. Okay? I'm not going to walk onto a facility and say, well, here's your problem. Your stocking density is 105%. I may look and say, okay, here's the constraints of the facility. Here's some other things. And I'm trying to think about it from a systematic standpoint. Okay, let's think about this from a management standpoint. What are the areas that we can try to improve and make some headway on this then, if we're seeing a high risk for infectious disease? Unfortunately, there's no silver bullet. I just don't know of one. You got to take that systematic approach. It appears both classical stress and alternative pathways are playing a role. I didn't get a chance to really get into stress. I think, some stressors are additive and compound on top of each other. And spreading some stressors out, it may be more beneficial, because we've seen some research where we can see behavioral changes in animals that's suggesting that these animals are stressed, but we don't see any changes in their leukocyte populations, or at least the functionality of the leukocytes we're looking at. And some of my conclusion is, yes, they're showing some indications of things that are potentially stressful, but we're not causing enough stress where we're tipping the balance in favor of increasing the risk for disease, because we don't see any changes in the leukocyte populations. We don't see any changes in the incidence for disease in those populations. It's not all about stress. There's alternative routes. I didn't get into too much about talking about negative energy elevation and ketones elevations and non-esterified fatty acids. I did mention how even subclinical hypocalcemia can cause a dysfunctional neutrophil response, and that may be associated with an increase for metritis in that study from Jose Santos's lab. And then lastly, it would be nice if we can identify cows that are at an increased risk of not adapting well and help them adapt. Cows are really good and animals in general are really good at hiding disease. We do a lot of disease challenge work in my lab, and that's one thing that's really became apparent to me, is that because we measure a lot of things in blood and other tissues after this challenge, and looking at these animals, they don't look like there's any clinical, we won't see clinical disease in some of our challenge models, but we see all kinds of things going on in terms of leukocytes and health, inflammation in these tissues of these animals. So again, trying to identify at-risk animals or animals that are off before we see clinical signs of disease. I'll kind of leave it at this. Once we see clinical signs of disease in an animal, we are way behind the 8 ball. So if we can find ways of identifying these animals earlier, we'll make some good headway. So again, thank you for the time, and I'm exactly at 6 o'clock, at least is what my phone says. I appreciate you guys listening, and would welcome any questions.
Excellent job. Okay, whilst everyone is formulating their questions, I please ask them to put them either in the chat window or the question and answer window, and I will read them. Paula will be translating the questions from Spanish into English, and Marcos will be doing that in Brazil from Portuguese into English. I'm going to take control back so that I can thank our sponsors and introduce the next webinar. So moving right along, let's see. Our next month, we will be joined by Dr. Heather Dan, a research scientist at the William H. Minor Agricultural Research Institute in Chazy, New York. She grew up on a dairy farm in New York where she developed a passion for dairy and an appreciation for research. Heather received a BS degree from Cornell University, an MS from Pennsylvania State, and a PhD degree from the University of Illinois. So you could say she worked her way west. Frequent listeners of the Nutritionist series will remember that Dr. Dan joined us to handle nutrition-based questions in the Dave Barbano talk on milk analysis last year. As we put together this year's roster, we asked her to give a full presentation on using milk analysis for real-time management decisions. Join us for the final presentation of the Nutritionist in 2017, and yes, we will be back in 2018, on November 8th at 5 p.m. Eastern Standard Time. Fun fact for those of you listening and confused in other countries and in the U.S., the United States will be returning to Standard Time next month. This messes up with our timing for the webinar. Because our co-hosting country, Argentina, does not change time for the summer, we've decided to maintain a constant webinar time for them. It makes it slightly confusing on our side and on the Brazilian side, but just remember to join us next time at 5 p.m. Eastern Standard Time, so a little bit earlier, but it always is different in February and November. I want to thank our people who make this possible: Tom Toluki and AMTS USA and Global, Marcos Neves Piera, University of Lavras, Paula Torillo in Argentina, and Marcelo Hens Ramos with 3R Lab, our translators at each location. We have generous sponsors who make it possible for us to get great speakers and manage the program. We thank our gold sponsor, Ajinomoto Heartland, Superior Nutrition through amino acids. Our silver sponsors are Arm & Hammer Animal Nutrition, R&D Life Sciences, Virtus, makers of Strata with EPA, DHA omega-3s, and Prequel with omega-6s, Dairy One Forage Laboratory, and Dairyland Laboratories. Our bronze sponsors are Jeffo, Life Made Easier, Adiseo, AminoMax, and Quality Liquid Feeds. I'm going to open up the floor now for questions, each for your English language listeners. Because we sometimes have problems with audio, please just type in your question either in the chat window or the question and answer window. I'm also going to open up the mics for Marcos and Paula, and I'll see who has the first question, because as yet I don't have any questions in my windows. So if you have any questions to ask of Dr. Ballou, please type them in. So Paula and Marcos, thank you for joining us. And Paula's busy translating. She tells me in the chat window. Paula, your mic is open?
Paula, tu micrófono está abierto.
Yes, thank you very much, Dr. Ballou.
Muchas gracias.
And we'll let Paula go back to translating. Marcos,
okay.
Do you have any questions in
Yes, I can you hear me?
Yeah, I can hear you, Marcos.
Oh, thanks, Dr. Ballou. Hi, Marian and Paula. Hope you're fine. There's a question here. What's the most important amino acid for immune function? You made a reference to that.
Can you say that again, Marco? Sorry.
Yeah, Marcos, I can't hear you very well. Can you speak up?
Can you? Is it okay now?
Let me see if I can. I'm worried that it won't come through for the rest of the people listening.
I'll try to. Can I write it? Can you listen to me now?
Go ahead.
Okay. What amino acid is the most important for immune function?
Oh, okay. The question is, what amino acid is the most important for the immune system? So I don't study this directly, but I do have a colleague here at Texas Tech that works with swine, and I know he's really, just based on conversations with him, he's really been looking at threonine levels, and he's, again, really focused on intestinal disease in this case. But again, it's going to be a lot of some of it may just be certain amino acids getting required in different concentrations. So you may see shifts in the different types of proteins being made you're going to require your amino acid requirements are going to change. So amino acid requirements during health are not necessarily the amino acid requirements during disease. And again, this is a pretty active area of research, but again, it looks like threonine is one that, in particular, that's been increasing or been shown the requirement's been shown to increase. But I would be speaking beyond what I know if I go beyond that.
Okay, thank you. I have another question here.
Go ahead.
In your opinion, what would be the most effective nutritional strategies or feed additives to increase immune function? What would you use first?
Yeah, okay, so that's a good question. So the question, I guess, is just so everybody, if they couldn't hear Marcos, is what types of feed additives would be maybe the most appropriate to improve the immune system. So again, it depends, and that's what, as academic people love to say, is it depends. And part of it depends on what disease we're looking at. So one feed additive may improve the health of, say, for example, reducing the risk for mastitis, where another feed additive may improve the resistance to gastrointestinal disease. So it's not as straightforward. I can get into a little bit of this. If you look at the different types of feed additives we have, and I'll kind of do more fungal right now, so I'll talk a little bit about yeast, and I'll talk a little about some other ones too. So we have various yeast products from yeast fermentation to autolyzed yeast which are more on the prebiotic side of things. Then you have some different extracts from yeast, so like a yeast cell wall. You also even have further extractions of yeast cell wall, so you maybe get higher contents of mannose oligosaccharides or MOS, or you may be getting further fractionating and getting higher levels of beta-glucan. And so again, looking at how these different yeast products work, some of them work more in terms of a prebiotic fashion, so they may be working with indigenous bacterial fungal populations in either the rumen or in the intestines in terms of a prebiotic effect. When you start looking at some of these extracts like yeast cell wall and specifically MOS are really good at binding gram-negative bacteria and binding other things. So we've done some stuff with that. And when you look at beta-glucan fractions don't bind well, but one thing that beta-glucan fractions do is they do stimulate some innate leukocytes and some other adaptive leukocytes as well. So you can see some stimulation or almost priming of leukocyte responses when you feed yeast cell wall or some extracts of beta-glucan. And I think there's been some data to suggest that some of those improvements in those leukocyte functions translates into improvements in disease resistance in terms of mastitis and maybe, I mean, most of our data is with mastitis and most of the data is done with mastitis, but you may also see some with metritis and respiratory disease. Gastrointestinal, you know, you get more into probiotics, and some of these yeast and prebiotics work with some of those natural bacterial populations there and stimulate certain bacterial populations to increase. And there's a lot of things going on there, and different strains of bacteria can do different things, either competing or producing various natural antimicrobial factors that are almost, I mean, essentially antibiotics or antimicrobials. So again, it really kind of depends on what you're looking at. I was talking with a friend of mine who's a scientist too, and he was doing some stuff with intravaginal probiotics, and he had mentioned he had seen some preliminary, some positive results there too. So again, it depends on the disease and that you're looking at, and kind of the strategy. I always say it needs to make biological sense what you're trying to do. So if you're having gastrointestinal, I'm going to look at strategies. Okay, what's causing this, and I'm going to look at strategies to reduce gastrointestinal. If I'm having issues with and again, it's difficult to know if you're having issues with neutrophil function, but if I'm having a high incidence of mastitis, it may be related to neutrophil function, and can I do something to maybe stimulate or recover some of that neutrophil function? So that's an area that my lab's is kind of looking at. Going back to looking at some of that data that we showed where neutrophil function, if it drops in the first, if it's suppressed at 7 days in milk, we see an increased risk for mastitis, or those animals are at an increased for mastitis, and they're also at an increased risk to get cold less than 28 days in milk. But if we also see animals that have elevated and it doesn't recover, then we're also seeing an increased risk that those animals are going to get cold. And in other data sets we have that show that those animals are at an increased risk to have decreased reproductive performance as well.
Okay, can I ask another one?
Yes,
Yes.
Go ahead, Marcos. I know Paula
Yeah.
has 3. I don't have any yet in my window, so go ahead.
And by the way, I will be leaving because I go to the Brazilian.
Right. Yeah, it's always good to get you done first.
Okay, so could you talk a little bit about the mechanism, how ketosis lead to low immunity in post-calving cows?
Yeah, so how does ketosis mechanistically lead to an increased risk for disease? So there's some evidence that ketosis directly, or ketone bodies can directly influence leukocyte function. So some initial data was done in vitro. You take some immune cells and you add some purified ketone bodies in culture, and then you stimulate these cells and you look at their activity. And adding low levels of ketones and NEFA, in that case, were shown to negatively impact functionality of a lot of different types of leukocytes. You know, another more recent study that was done that it was a very nicely done study. They infused ketones into live animals, and I'm trying to remember if they did a challenge or if they just took cells and looked at their functionality. But what they saw were those animals that they had infused ketones. Again, these animals were in a positive energy balance, but they infused ketones at a level that these animals were that a clinically ketotic animal would be at, and they showed decreased. It was either decreased leukocyte responses and or decreased resistance to a disease challenge. So there is appears to be some direct effect of ketones. Also I think, I was jumped on early and I was talking to Lance or listening to Lance's presentation, and I thought he made a good point about talking about ketosis in cows, and that, I want my animals to have some degree of ketosis in their lactation, because that means they're mobilizing body fat and they're putting a lot of that energy into milk production. But I want animals that are also consuming a lot and they look good. And so, again, I think some of this high levels of ketosis and negative impacts of disease may be indirect also, or there may also be some indirect effects taking place where in the part that's causing the negative impact on the immune system, the risk for disease may not be directly associated, or the indirect aspect is also contributing to the impaired resistance disease. I don't know if that makes sense.
Okay, thank you.
It sounded good in my head, so I don't know if it came out as well.
Oh, that's great. Thank you. Thanks a lot.
All right, thank you, Marcos and Marcelo. Thank you for joining.
He can translate it into Portuguese, I guess.
Yeah.
We'll try.
Yeah, we will see you next month. Thank you for joining us.
Thank you.
Dr. Ballou, I appreciate you pulling.
Un silencio ahora.
Hi, we cannot hear Marian. Dr. Ballou, do you hear us?
I can hear you, yes.
Okay, I will ask you a few questions while Marian
Le voy a hacer algunas preguntas mientras
enters again, okay?
Okay.
vuelve a conectarse.
Okay, the first question is from Pedro. How do you explain that dominant cows, having feed to eat, get more stressed than submissive ones? One would think that dominant cows, being more aggressive, would be stronger.
Porque uno pensaría que son más agresivas.
That's a great question, and you know what's interesting is if you get outside of dairy, we talked a lot about subordinate cows, and there's a lot of data on subordinate cows having an increased risk for disease. But if you look at other animal models, and one of my favorite authors on stress is a professor at Stanford. He actually studies baboons in Africa. And it's funny when he looks at his population of animals and who's most likely to get sick, it's those dominant animals. And his thinking behind it is they're chronically stressed. They're always trying to stay on top, and animals underneath them are always trying to test that. And so they just can't relax. And so that's kind of what our thought process is you have some dominant animals, but those overly dominant animals that are just constantly engaged in dominant activity are the animals that were in that data set with Ricardo Chabel, were the ones that were at higher risk for metritis, and then subsequently also a higher risk to be removed from the herd. So again, we think it has more to do with, yes, some dominance is good, but too much dominance is a bad thing when these animals, and again, we were just hypothesizing in that data set that those overly dominant animals actually aren't eating. But I also think there's more to it than that. I think those overly dominant animals, they're also just concerned with trying to stay on top. And that's consistent with some other animal models, like I had said with those baboons, which I just think is kind of fascinating.
Okay, great. The second question is from Gustavo. You talked about the effects on macrophages and neutrophil function, but humoral immunity is important in this period?
Yeah, I didn't really go too much into humoral. I didn't go really into humoral immunity at all today. Yes, having some humoral protection is definitely important, and we know that a lot of the lymphocyte responses can be suppressed in early lactation and are suppressed in early lactation. Specific to mastitis, again, if there's a pathogen leukocyte interaction there. So I had mentioned that we're designing a more comprehensive study to look at different leukocyte populations that may be associated with negative health outcomes. So that one study that we did, we looked at just neutrophil functionality in that study, but we want to look more at what is the association that we're seeing with some of these suppressions and different lymphocyte or humoral type responses as well. So yes, they do play a role. yes, we know that a lot of lymphocyte responses, specifically T cell responses, certain T cell responses are suppressed or can be suppressed in early lactation and probably contribute to the increased incidence of some diseases. So yeah, that is a good point.
Okay. And now, this is a long question from Pedro. Which would be the measure to have or to make to detect animals prone to having stress, just to be earlier than clinical expression? As you said, we are always late regarding the problem.
Yeah, that's a good question. So I'm going to approach this two ways. So the question was, is there a strategy that we can identify animals that maybe are more prone to stress? So to answer that, yes, I think there are some things we can do. I do some work with, I live in Lubbock, Texas, and if you don't know that, it's the feedlot capital of the U.S. And so I do some work with receiving cattle and feedlot cattle. And one thing that we've done some stuff with is looking at temperament of animals. And so again, looking at chute scores, how fast, the exit velocities, how fast an animal leaves the chute. We've also do some approach scores where we go into a pen with an animal and we assess does the animal want to stay away from you? Are they highly excited? And we classify these animals as low, moderate, or highly temperamental. We've done some different things looking at the effects of different stressors on animals of different temperament. And like you would think, these animals that are more temperamental have a higher or have a more profound impact. Stress has a more profound impact on their immune system or leukocyte responses than these more calm animals. So even just doing some things like that, looking at temperament of animals, submission versus dominant. So if you have overly dominant animals, and I think you can see those animals, we know that those animals are not only going to have a higher risk for disease themselves, but they're also creating probably some issues with some other cows in that pen. So in terms of stress, I think doing some visual assessments of stress can do that. In terms of disease, identifying disease early, I'm excited about it because typically we'll see changes in behavior before we see changes in clinical signs of disease. So a lot of the stuff looking at feed intake, rumination collars, I think there's a lot of potential benefits there in terms of identifying animals that are off. Okay? They're deviating, looking at deviations from the normal activity. Something's going on. So I always say if an animal you think looks a little off, they're probably off because by the time they're even just showing a little bit of something going on, the stage has already been kind of set, it's been put in motion. So I hope that answers your question.
Yes, I think so. And I have another one related to this last question. Could we think about genetic selection to animals less prone to being stressed?
Yeah, I know that, back to my temperament deal, I know that there's bulls and they've done some stuff with heritability of some of these traits in terms of just temperament of animals. And so, understanding that. And actually, if you ask feedlot guys, what would be a genetic trait that they would like? They like calm animals. And so if we can find traits and breed for, less temperamental animals, I think they'd be happy. I don't know of any evidence of where people have looked specifically off the top of my head, looking at the heritability of maybe a stress-related. But we know that there are some disease genotypes that are and even just using some SNP chips, that there are some polymorphisms that make animals more resistant to infectious disease as well. So I think using the genetic component is going to play a role. But even if you look at genetics, our environments are playing such a big, are contributing so much variation to disease incidence and just even when I look at certain leukocyte responses, and I try to contribute it just say to herd level effects versus within herd level, and I'm looking at a specific leukocyte or a group of leukocyte responses. Herd contributes to leukocyte responses, so generally if I just throw a number out there, I would say it contributes 10 to 15% of the variation in my data sets. But again, that 85 to 90% of the variation is still within a herd. So again, genetics I think plays a role, and even management factors play a role, but there's a lot of within animal variation on any facility you go to.
Okay. Can you hear me okay now?
Yeah.
Good. Paula has another question, but she needs time to translate it. I do not have any questions in my chat window or in my question and answer, but I did want to just say I appreciate that you referenced Dr. Baumgart's talk from last month, because one of the things I try to do is set up these webinars as sort of series. And our next webinar cycle in 2018, we're going to have Chris Chase speaking on more immunology and nutrition. Interestingly enough, Elliot Block he suggested that I talk to you and also to Chris Chase to do an immunology series. I do have a question, so while Paula is translating, I'm going to go ahead and ask that. This is from Carl Cass. He asks, could higher incidences of UI in dominant cows be because of the self-influenced stress on the cow, just like humans who are under higher stress from always wanting to be first tend to have higher rates of heart attacks? They have a higher level of stress even under normal conditions. What's your thoughts on that?
That's the simplest answer. I think that's exactly what's going on. So those animals are more responsive, I think, to stress, and the fact that they're always just trying to stay on top. They're always trying to maintain their dominance. There's a higher incidence of disease among those animals, and I think that's exactly correct.
Okay, thank you. I'll see if Paula is ready.
Marion, just don't tell Lance that I referenced him. I don't need.
Oh, okay. I won't.
I don't need him holding that over my head.
Very good. Okay, Paula's ready with her question.
Okay, I'm ready. The question is from Gustavo. Is there information regarding those vitamins and minerals you talked earlier, injecting them during dry pre-fresh periods and at freshening to make better immune responses or to lessen the oxidative damage?
Yeah, so a lot of those trace nutrients I talked about, I kind of singled in on a lot of the ones that are more, they can act as antioxidants, but sometimes they can also do some other things. And there is a lot of data to suggest that, again, you can deplete those antioxidant systems, and anytime you shift the balance in favor of the oxidants, you increase the risk and you increase the degree of oxidative stress, which can have some negative impacts on the immune system. When you specifically look at individual, and again, these antioxidant systems, there's a lot of synergy in those as well. Selenium and vitamin E is a great example of that. When you look at, you try to isolate one specific one and talk just about that or study just that, it gets a little complicated, and really you have to see a deficiency in there to really start seeing negative impacts on disease resistance. The vitamin E was kind of a unique one that we saw it increase in 2001 recommendations to such a degree, but that was because there was a lot of data supporting both improvements in neutrophil function, but also downstream of that, improvements in resistance to mastitis. When you look at supplementing higher levels than current recommendations with a lot of those micronutrients, you usually don't see much of an improvement unless they're under a majorly stressed situation where the oxidant production is a lot higher than we would typically see. So I suspect that most of those recommendations aren't going to change much.
Okay, I have a question from Tom, and he asks, are there going to be major changes in mineral nutrient feed guidelines in the NRC 8 compared to NRC 7 for dairy cows? Do you know?
I do not know, and I am actually very interested in knowing that. And that's why I was cautious, and I probably shouldn't have said it, but because I may, when it comes out, there may be all these changes in these micronutrients, but I suspect, with maybe the exception of manganese, there's not going to be a major change.
I know that for next year we have Dr. Weiss scheduled, and I'm hoping to get him late enough in the year that the NRC will have come out.
Exactly, because he won't talk about it probably until he can. So.
Yeah, so maybe I need to schedule him in November the way things are going.
Yeah, that would be an interesting talk.
Yes. Okay, I think that we are set for questions unless I get something quickly. But I do want to thank you for joining us. I appreciate your references to human issues as well. I as a mother, I always felt frustrated with my knowledge being much deeper about dairy cattle than it was about human beings, and they seem very nebulous on giving me lactation information for people. I just applied my cow knowledge. It worked.
Yeah, no, I go back and I use both, and like I said, I had read a lot about this baboon data, and I just thought it was fascinating. Then I look at dairy cattle literature, and we always talked about the subordinate, and I'm like, what about these dominant animals? And so then we did this study, and Ricardo was sharing it with me. It kind of fit along some of my hypotheses. So,
yeah, it's far more complex than we even imagine. So
yeah. Well, thank you again for inviting me.
Paula says thank you as well. So.
All right, you guys have a nice evening.
Thank you. You too, and take care of your cold.
I will.
All right.
Thank you. Bye.
Thanks very much. Bye.